Hi, everyone. I'm Sydney Harned. I am a PhD candidate in the Burford Rice-Kind Lab at North Carolina State University. And I'm excited to talk to you about some of my dissertation work looking at the fine-scale population structure of juvenile southern flounder in North Carolina. So some quick background on these fish and why they matter. Southern flounder are found in the southeast Atlantic from North Carolina down to central Florida. There's a break around South Florida, and they're found in the Gulf of Mexico. Southern flounder are an important fisheries species. They're targeted by both commercial and recreational fisheries, and in North Carolina specifically, they're considered one of the most valuable finfish. So as you can probably guess, with a highly targeted fishery species, southern flounder populations are declining. The black trend line shows the minimum spawning stock biomass for sustainable harvest of these fish, and the gray trend line shows where we are in reality as of 2017. And projections show that these populations will continue to decline if we continue on the current path, with extinction potentially as early as 2046. And the population declines of southern flounder are exacerbated by temperature-dependent sex determination in these fish. So at suboptimal temperatures or when exposed to other environmental stressors during a critical period of development, genetically XX female flounder will develop into phenotypic males. So that means they look like males, they act like males, they can reproduce as males, but they have two X chromosomes. And this is a problem given that female flounder are much larger and grow faster than males do, so they are disproportionately targeted in fisheries. So when you consider that fewer females are being produced and more females are being taken from the environment in adulthood, that spells disaster for this species of fish. So there have been some studies of population genetics on this species of flounder in the past, and we know that there are two distinct populations or stocks in the Gulf of Mexico and the Southeast Atlantic. However, the previous studies didn't find any significant genetic structure within these ocean basins. You can see in this admixture plot at the bottom of the screen from Anderson et al. 2012, the Gulf of Mexico and the Southeast Atlantic look very different from each other, but other than this small bit of differentiation, the Southeast Atlantic looks pretty much genetically the same. However, it's important to point out that these studies were done using some older methodologies, including mitochondrial DNA sequences, microsatellites, and allozymes. And while these methods are useful and informative, population genomics and high-throughput sequencing increases the power of the data set and may allow us to detect more fine-scale population structure that may have been missed using those other methods. So the question I'm asking is, can we detect fine-scale population structure within the Southeast Atlantic using high-throughput genomic sequencing? And to answer this question, I used reduced representation genome sequencing, ddRAD-seq, and I was able to generate 154,808 SNPs over 3 different sampling years. And with this huge increase in marker number, we have better power to detect fine-scale structure. So I chose to focus on juvenile southern flounder found in North Carolina. Southern flounder adults spawn offshore, typically in the winter or spring, and the juveniles have a 30 to 45 day larval stage where they're hanging out in the water column before they settle in bays and river basins and estuaries after 45 days post-hatch. And these fish will remain there for 1 to 3 years before reaching maturity and moving offshore. So the map on the right shows the specific sample locations that we had. I had a total of 207 individuals over 3 sampling years. 2015, 2016, and 2022. And while the specific locations might change a bit from year to year due to sampling constraints, the three broad locations we looked at were mostly consistent, and those were Pamlico Sound in the north, the Neuse River, and the New River in the south. So now we can get into some of the preliminary results that I'm finding. The graph in the bottom left is a DAPC. DAPC is a multivariate clustering method that aims to define genetic clusters and plot the relationship between these clusters. And you can see that even in this very high gene flow species, we are seeing pretty separate clustering of these a priori locations. However, we did not find pairwise, significant pairwise FST values between these locations. And now we can look into the 2016 year. So you can see from this DAPC that even though we are still seeing some clustering, there's a lot more overlap happening between these locations, and the separation isn't as dramatic here. But what is interesting is we did find significant pairwise FST values in this year. So the circled locations show the three locations that were significantly differentiated from the other sites that we sampled. And those were Swan Quarter, Hancock Creek, and Mill Creek. And finally, we also looked at structure in 2022. Due to sampling constraints this year, we only had 2 locations, and both of these were located in the Neuse River. The salinity this year was higher than expected, so the juvenile flounder were higher upstream than the boats were able to reach. So we only have these 2 sampling locations, but even with 2 locations, we're still seeing this structure show up, and they're with 2 locations that are located pretty close to each other. So the DAPC shows that with some overlap, there's still some distinct clustering, and the FST value was significant between these 2 sites. So I want to bring it back to our 2015 and 2016 DAPC results, because I think what we might be seeing here is temporal pulses of juveniles from separate spawning cohorts. So it's important to look at the timing of when these samples were collected. The samples in 2015 were a bit more disjointed in their temporal sampling. All of the locations had samples that were collected in May. However, the circled locations and the ones that showed the most distinct clustering from the others included samples that were collected in June. So we have seen in this species that the river basins and estuaries will have increases in juvenile abundance throughout the season in what seems like pulses. So we think we might be sampling these pulses and seeing differences in the offspring of adults spawning at different times throughout the season. In contrast, in 2016, our sampling was much more even, and we had a more homogeneous mix of samples that were collected from May throughout July. So here we think we might have sampled all of the pulses rather than having the separation, which could be why we aren't seeing as distinct clusters here. So in conclusion, we did find genetic structure both within and between river basins in North Carolina. This is exciting and novel given that previously the Southeast Atlantic was considered a single admixed stock. So we're seeing that there is a lot more fine-scale structure here than we expected. We found that genetic structure is variable among years. And again, this could be due to pulses of juveniles from adults spawning at different times throughout the season. I found inconsistencies in the measures that we used. The pairwise FST results don't always match up with what we're seeing in the DAPC. And I'm still actively working on this study, so I don't have an explanation for that yet. If you have any ideas and you want to come talk to me afterwards, that would be awesome. But I'm still trying to figure out biologically what might be happening here. But overall, we're seeing that stock structure is a lot more complicated than we had originally thought. And more studies using larger marker sets and high throughput genomic sequencing are needed to tease out the fine scale structure that we're seeing within ocean basins. So the next steps of my work, first and foremost, I am looking at the temporal genetic differences among samples sampled at different time points throughout the spawning season. We found these interesting discrepancies in the timing of the samples, so I was able to work with the North Carolina Division of Marine Fisheries and collect juveniles at different time points ranging from April to October in the same locations used in this study. And I'll be looking at population genomic differences between these different time points to see if this hypothesis of sampling these pulses holds. I'm also working on assembling a fully phased whole genome for both summer and southern flounder. We are working with Virginia Tech, Holly, who I see, to assemble a fully phased genome and investigate the effects of temperature on sex determination in both of these flounder species. So currently only a genome exists for the Japanese flounder. So we'll be assembling this genome in both males and females. So ideally, we can identify genetic differences in the sexes and look at how prevalent temperature-dependent sex determination and sex reversal is in these species. And with that, I want to thank Martha Burford-Reiskin and the Burford-Reiskin Lab, the North Carolina Division of Marine Fisheries, who helped acquire the samples for this study, our undergraduates that helped out with the lab work and the funding sources, the Morgan Family Charitable Foundation, and the GGA Seed Grant. And with that, I think I have time for maybe a couple questions. Yeah.
Such a cool talk. I was wondering, I'm not familiar with the DAXI method. Does it take into account outlier loci?
That's a good question. I believe it does. It's not looking at neutral loci like pairwise FSTs are. So that's something I was actually looking into last week, seeing if maybe some of these differences are due to loci under selection. Right, yeah, so that's something I'm looking into. We might end up running Locitan for these and seeing kind of if there's differences there. But yeah, that's definitely a possibility.
What do we do in that question? Do you know if there is variation in the environment in the different ones?
Differences in environment?
Like salinity or temperature?
Definitely, yeah. So some of our southern locations are much warmer than the northern locations. And a previous study done by some of our collaborators actually found really significant sex skew in some of those southern New River sites. So we could be seeing differences there because of temperature adaptation, thermal stress. So that's something else that we're looking into as well. Anyone else? Mm-hmm.
Great talk.
Thank you.
So you showed some of the pulse results.
Mm-hmm.
There was variation in time, sampling of time impacted some of the genetic structure. What about between years? Right? Was there temporal variation in structure between years?
That's a great question. I need to run my analysis with all of the years. I did that, and it was a little bit hard to tease out what was going on because there were just so many sampling sites. But I think, yeah, one of my next steps is going to be taking the locations that we have repeated in years and looking at those separately and seeing if we're seeing differences that way. Yeah. Cool. Thank you.
All right, hey everybody. My name is Blair. I am a postdoc at UC Santa Cruz with Joanna Kelly, and today I'm going to give you kind of a high-level look at some ongoing work we're doing to study gene regulation, physiology, and hibernation in brown bears. So, hibernation is probably something everyone's vaguely familiar with at least. This is an adaptation that enables survival during prolonged periods of resource scarcity, most commonly associated with the winter season. In mammals, this is comprised of a suite of physiological shifts that occur during this hibernating period, including reductions in metabolic rate, reduced body temperature, limited feeding or complete fasting during this time, limited activity, and a lot of pretty interesting and often medically relevant shifts such as temporary onset of insulin resistance in various tissues during hibernation, the maintenance of muscle tissue despite little to no movement during months at a time, that make this a pretty cool and relatively complex system to study. And as you can see here on the right, it's kind of shown in blue. Hibernation really pops up throughout the mammalian tree, is not restricted to a particular clade, which also adds some interesting evolutionary dimensions to the study of this as well. But most of my work at the moment is focused on hibernation in bears, which are maybe one of the kind of most charismatic examples of this. Bears spend roughly half of their year hibernating, so typically about November to March will be spent hibernating, and as they emerge, they essentially start preparing for the next season of hibernation. So during the summer and then the fall season, they mate. They essentially focus entirely on gaining as much weight as possible in preparation for the winter season, as modeled by Holly over here on the right. These pictures were taken the same year, July and September, gaining as much weight as possible so that they can use that adipose tissue to sustain them throughout the hibernation season. So as you might expect, these complex and kind of high magnitude physiological shifts are driven in part by massive shifts in gene expression. So in the adipose tissue, where really a lot of this kind of metabolic activity is shifting, we see over 6,000 genes that are differentially expressed between the active season in summer and hibernation in winter. Liver and muscle, other tissues we've looked at, we have 2,000, 5,000 genes. As you might expect, a lot of these are involved in metabolism. So in adipose in particular, we see shifts in insulin signaling genes, lipid metabolism, other metabolic pathways that are more specific to liver and muscle in those tissues as well. And another thing that makes bears, I think, a particularly interesting system is that, kind of similar to the mammal-wide tree, hibernation is variable within bears. So this is just 4 of the 8 currently extant bear species, kind of the most well-known ones. Not all bears hibernate. So panda bears do not hibernate. American black bears and brown bears do hibernate. And polar bears, it's complicated, but essentially they don't hibernate. I'll talk about a little bit more about that here in a second. But they're a particularly interesting case here because the estimated divergence between polar bears and brown bears is only about 500,000 years ago. So in a relatively short period of time, there's been a pretty substantial degree of metabolic and physiological specialization and adaptation in the polar bears in particular. Again, no real annual hibernation. They've shifted to a fully carnivorous diet. They have high resting metabolic rates compared to other bears. And they really experience unique metabolic and energetic demands associated with this Arctic marine lifestyle. So I'll come back to this a little bit later. But more generally, my work and some of the other work in the Kelly lab is interested in understanding the mechanisms, regulation, and evolution of hibernation in bears, and then scaling up across mammals as well. So what are the underlying genomic mechanisms of this? How is it regulated? How are we seeing, what is controlling these massive gene regulatory shifts that we see between seasons? And kind of putting that all together to try to address how hibernation has evolved in bears and in some of these other mammalian lineages. So for example, do we see convergence in these distinct lineages of mammals that do and do not hibernate? And something I'm going to focus on particularly today is the role of regulatory sequence evolution in the evolution and kind of gain, loss, and modification of hibernation across bears and mammals. So for folks who maybe haven't thought about gene regulation in a little while, one important aspect of gene regulation in eukaryotes is cis-regulatory elements. These are parts of the genome that are bound by transcription factors to essentially control the how, when, and why of gene expression. Most common are promoters, enhancers, silencers, and insulators. And today I'm going to focus primarily on enhancers, which are often located 10 to 100 plus kb away from the genes that they target. And through these chromatin loops come into contact with the promoters of their target genes to control, again, kind of the context, timing, et cetera, of gene expression. And why I think regulatory sequence evolution is really interesting, and particularly for this system, is that by changing these regulatory sequences, you're able to modify phenotypes by altering either the expression of particular genes or potentially kind of bigger changes in the way that these genes interact with one another, kind of rewiring or modifying these regulatory networks. So essentially, in other words, you're tweaking a gene's activity in particular contexts, whether that's a tissue, time point, et cetera, while preserving its activity in others versus, protein coding changes are essentially going to be universal throughout the entire organism. And because of this, enhancers in particular have been shown and kind of theorized to evolve rapidly in a lot of different cases and contribute in a lot of situations for, or in the adaptive evolution of complex, multi-gene expression-driven phenotypes, perhaps hibernation, right? So a lot of our work is starting to try to both uncover the regulatory landscape of hibernation in bears and then use that to start to try to gauge the role that regulatory sequence evolution has had in metabolic and physiological adaptation across bears, for example. And I'm going to talk about both of these a little bit today. So starting here, we first wanted to identify enhancers in the bear genome that are, involved in metabolism, hibernation, et cetera. And we immediately hit a roadblock, which is that adipose tissue is kind of the most interesting tissue in a lot of cases when studying metabolism associated with hibernation. And it's also essentially the hardest tissue to generate a lot of the functional genomic data that you would typically do to identify enhancers. So ATAC-seq data for open chromatin, ChIP-seq data for histone marks, and so on. Essentially, the fatty nature of this tissue just makes it really, really difficult to do those sorts of techniques. And while they have been done in mouse and human and so on, turns out they don't work very well for these non-traditional models like bears here. So we wanted to try to find a detour around this so that we could keep making progress. And we landed on something that's kind of a weird but I think pretty cool solution, which is this relatively new approach called cap small RNA sequencing, or csRNA-seq. So this was published about 5 years ago. And this method was really developed to be able to really identify and map out transcription start sites in the genome at a really high resolution, single nucleotide resolution in some cases. But it does this by essentially capturing nascent RNAs. So these are RNAs that have just recently been transcribed. They're small, they have a 5' cap. And what's cool about this technique compared to traditional mRNA-seq is that you capture stable RNAs, so like mRNAs, non-coding RNAs, and so on, but also a variety of unstable RNAs, which include these enhancer RNAs that I'm going to talk about, which are really key for us here. And these unstable RNAs typically get degraded pretty quickly. You're not going to capture these in typical mRNA-seq, for example. And we can actually use these enhancer RNAs to identify enhancer sequences. And what's really valuable for us here is that this uses total RNA as input. So you don't have to go through these more involved protocols like ChIP-seq. If you can extract RNA, you can essentially do this protocol. And it makes it a lot more feasible for things like adipose, which we can get RNA-seq data from. So real quick, without going into a ton of details about this approach, I did mention that this captures these enhancer RNAs, or eRNAs. These are short, unstable transcripts that, the function of these are still kind of up for debate in some cases, but a lot of times they're thought to essentially result as almost a side effect of these open, accessible enhancer regions. And typically they kind of just get degraded and you don't really see them in your typical RNA-seq data. But this approach actually captures these before they're degraded, and similar to how you would map mRNA-seq back to the genome to identify protein coding regions, we can map these transcripts back and try to actually identify the enhancer regions that these enhancer RNAs are coming from. A couple useful features of eRNAs from previous studies is that they correlate with enhancer activity. So if enhancers are active in a lot of cells in your sample, you're going to find more eRNAs from that enhancer. And they also kind of correlate expectedly with the expression of the genes that these enhancers are targeting. So we kind of can use these to start to associate these enhancers with target regions, or target genes, rather. We generated this csRNA-seq data and some associated mRNA-seq for adipose tissues from bears during both the active season and the same bears, again, during hibernation. Real quick, I want to mention that this was done at the Washington State University Bear Center, which is a captive population of brown bears. Allows us to take these samples safely and non-invasively, and is pretty much the only way that we could do this work. So it's a really special and one-of-a-kind place. But using this csRNA-seq data from these two different seasons, we were able to identify putative enhancer regions, mapping this back and specifically focusing on regions where the csRNA-seq data was mapping to these distal regions, so far from protein coding genes. And these transcripts were identified as unstable, so kind of the expectation we have for eRNAs coming from enhancers. And as this is just kind of a weird version of RNA-seq, you can do like differential expression analyses like you would typically do with mRNA-seq data. So we can further focus in on these enhancer regions that have evidence for actually changing expression or really enhancer activity between the active and hibernation season, which we were able to do here. And as I mentioned, eRNA abundance correlates with the target gene, so we're still kind of finessing this, but essentially by correlating the change in eRNA abundance between seasons with nearby differentially expressed genes, we're starting to kind of, associate these enhancers with the genes that they supposedly target. So in addition to just identifying maybe enhancers in the genome, we've been able to focus in on ones that we think are specifically either, driving or responding to these metabolic shifts associated with hibernation. So, this is kind of basic, but this is pretty exciting for us because this gives us a set of really interesting cis-regulatory regions to start to investigate, for example, to investigate the roles of regulatory evolution in this system. So I'm going to talk about really just kind of the first steps of this, or where we're at currently, which is early stages of this in the few minutes I have left. So as I mentioned before, I think one of the most interesting kind of stories or questions within the system is the high degree of physiological and metabolic adaptation that's occurred in a really, really short period of time in the polar bear lineage compared to the closely related brown bear. So that's kind of what I'll focus on the context of this here moving forward. When we looked at the orthologous regions for these putative enhancers across the bear genomes here, we found that generally they're pretty highly conserved, which is something you tend to see and tend to expect for regulatory regions like this. But to the extent that when we look at brown bear and polar bear, 65% of these were 100% identical pairwise sequence identity between these species. But when we look at the ones that are not completely identical, we can look for evidence of positive selection, which we did through this approach called Adaptify. As these are non-coding sequences, we can't do like DNDS, et cetera. So this approach and similar approaches use estimates of substitution rate, looking for an elevation of substitution rate in these enhancer regions in our case. And a little underwhelming at the moment, but we were able to identify some enhancers with evidence of positive selection, and pretty modest numbers though. And when we focus in on the second row here, on enhancers that we have that differential activity inference from the brown bears in particular, so ones we think are really involved in hibernation and metabolism, gets even smaller with only 7 under positive selection in polar bear. Real quick, there are some genes that we infer to be targeted by these that we think are really relevant with roles in adipose metabolism, insulin sensitivity, and so on. But there's not really any obvious surplus in the polar bear lineage compared to the other ones. So at the moment, there's not some magic obvious signal that there's a really strong role of the regulatory sequence evolution in this lineage in particular. But I just want to mention that this is one of a large number of axes of potential regulatory divergence that could contribute to this, whether chromatin accessibility we haven't looked at, transcription factor activity, higher level signaling, and so on. So there's still a lot that's left to be unknown, so hopefully we'll have some more exciting answers next year at Evolution. But we're doing a lot of work that I don't really have time to focus on, but I'd be happy to talk more with people about afterwards. So with that, I would like to acknowledge the Kelly Lab at Santa Cruz, some really awesome undergrads at Washington State, everyone at the Bear Center, and funding sources, and I maybe have time for one question, but here's my contact info, so reach out, find me afterwards, and I'd be happy to chat more, and thank you all for being here. Maybe one super quick question before I get my gong. All right, well, thanks a lot.
Hi, everyone. Thanks so much for being here. My name is Hannah Aichelman. I am a postdoc in Sean Mullen's lab at Boston University, and I'm really excited to tell you about my project investigating the genomic basis of mimetic color pattern variation among hybridizing populations of these really beautiful admiral butterflies. So research in the Mullen lab is really focused on understanding kind of origins and maintenance of diversity across multiple levels of biological organization. And we think a lot about butterflies in the context of these sorts of questions because they're not only really beautiful, but there's a ton of diverse, and they serve really interesting and complex functions. So these butterfly wings can play, and the patterns that are on them can play roles in everything from thermoregulation to mate choice. Just a few other examples include kind of warning signaling, like aposematism in the top left, crypsis, the ability to blend into the background and avoid predators, and also mimicry. So
we think a lot about Batesian mimicry, and I will be telling you a story about this type of mimicry today. But this involves a palatable mimic species that's protected from predation because it resembles a toxic model. So that's shown here in the top right, in these Papilio butterflies. But the Batesian mimicry complex I'm going to tell you about today includes these 3 butterflies. So on the left, we have Adelpha californica, the middle is Limonitis lurkwini, and on the right is Limonitis whitemeyeri. And Adelpha is our toxic model. These butterflies eat oak, and they sequester the tannins from the oak. These tannins are unpalatable, and it makes them toxic to predators. And Limonitis lurkwini is a Batesian mimic of Adelpha, produces this really conspicuous and pretty orange patch on its wing. This is called an apical forewing patch. I'll also call it an AFP for short. And additionally, Limonitis lurkwini exists in symmetry with the sister species, whitemeyeri, and when they're found together, they hybridize freely. And when they hybridize, they produce a really wide diversity of extent of this AFP, this orange patch, from more lurkwini-like, a higher extent of this AFP, to more whitemeyeri-like, or much less of that AFP. So this, the parent species here, Limonitis lurkwini and whitemeyeri, do still vary in the presence or absence of this really conspicuous patch, this AFP. So this suggests that divergent selection from mimicry could be playing a role in maintaining the species boundaries between these 2 butterflies. And so some previous work by a PhD student in the Mullen lab found some really nice evidence of this. This is from a paper that Evan Christensen published in 2018. And so essentially he found a few key findings that suggest to us why Batesian mimicry is important in this system. First, he found that Limonitis lurkwini experiences reduced predation when it exists in symmetry with Adelpha californica, this toxic model. Additionally, the mimetic color pattern variation, so this AFP, is more variable in allopatry. So when lurkwini is away from its toxic model, the kind of extent of this AFP is more variable. Additionally, there was protection for lurkwini that eroded when it was away from the models. And finally, a mark recapture study that Evan did showed that the mimics survive a lot longer when the models are absent. And I should say that he did this with a massive amount of fake butterflies that he put out in the field along the range of these species, which is really amazing. So all of this together suggests to us that selection for Batesian mimicry is the cause of this convergent phenotype, this orange apical forewing patch. But we still don't know very much at all about the genetic basis of this phenotype. So that's the objective of my work, and the question that I'll tell you about today is kind of what are the genes that are underlying this mimetic phenotype, this AFP, in Limonitis lurkwini. So to ask this question, I have 2 main objectives. First, to assemble and annotate a new Limonitis lurkwini genome, and this allowed me to get to objective 2, which is to conduct a genome-wide association study to identify the variants that are associated with this AFP. So for objective 1, our Limonitis lurkwini genome assembly and annotation, we had PacBio hi-fi reads, which I assembled with hi-fi ASM, and then purged duplicates with purge haplotigs. And because we don't have any transcriptomic data for this species yet, I annotated this genome using a program called Liftoff. So I was able to take annotations from a closely related species. 2 in this case, I used Limonitis camilla and Heliconius melpomene, and I was able to use those annotations to infer annotations in our genome, which ended up working out quite well. And then I used Eggnog Mapper to decorate my annotation file that I got out of that pipeline with Liftoff. So our assembly turned out pretty well. We have 64 scaffolds in our assembly, a total of 360 megabases, and an N50 of 12.95. And it's quite complete, so we have 97.7% complete single copy orthologs in our genome. So we were really excited about this. And with this nice genome, we can now move on to our second objective. So using whole genome sequencing to conduct a genome-wide association study, or GWAS. And to do this, we had multiple sampling efforts that we combined into one analysis. So I'll tell you a little bit about that now. We had first 96 F2 and backcross individuals from a mapping brood sampling that and additionally we had 52 wild-caught individuals, which included our two parental species, Laminitis lorquini and Weidemeyeri, as well as some wild-caught hybrids. And after sequencing and removing individuals that didn't have enough coverage, we were left with the sample size, so 91 F2 and backcross individuals, and then 51 wild-caught individuals with pretty even sampling across our two parental species and the hybrids. I conducted GWAS using GEMMA, and for now we're just considering our AFP phenotype, our wing patch phenotype, to be binary. So 0 if you have a black wing or no AFP, and 1 for any extent of the AFP. I'll tell you a little bit more at the end about what we're going to be building on this data set with moving forward. But for now, some data. So here's our genome-wide association study with all of our samples, and we have a really broad scaffold-wide association with this phenotype, a little bit like it's flipping me off. But we think that what's happening here is that we're including our mapping brood individuals in this GWAS, and so we just don't have a lot of recombination in these mapping brood individuals, which is resulting in this really scaffold-wide association. So to try and address this, we reran the GWAS, but just with our wild-caught individuals, and so that's what I'm showing you here. This is now wild-caught only individuals, and we're zooming in on that scaffold 13, our middle finger scaffold from the previous slide. And now you can see that we're starting to get a more clear, narrow signal on the association with our AFP phenotype, highlighted here in these green points. So we want to know a little bit more about what's happening on this scaffold 13. So now we're going to zoom in even a little bit further. These green points are now represented by the same extent of this scaffold here highlighted in green. And the first thing that I want to point out to you is that the scaffold-wide FST on scaffold 13 is quite a bit higher than the genome-wide scaffold. So it seems like potentially some evidence for selection on this scaffold, which is exciting to us. Additionally, when we looked a little bit further at what's going on here, we found 12 fixed SNPs that are right inside this region of high association with our AFP phenotype. And when we looked at what annotated genes were in this region, we found a really exciting one. So it turns out that optics, a really well-known color patterning gene in Heliconius butterflies, is also here, right in this region of interest to us. And so what we think is happening, and that we're going to kind of continue working on, is that potentially these 12 fixed SNPs here are involved in regulating the expression of optics to produce this AFP phenotype in our butterflies. And again, this is really exciting because optics has been previously shown to be a really important determinant of similar kind of red and orange wing pattern phenotypes in these Heliconius butterflies. But these Heliconius and Laminitis are diverged by about 65 million years, and so this is a cool finding that the same gene is involved in our system as it is in Heliconius. But we have some more work to do to confirm the role of optics, so we're following up this GWAS with some ATAC-seq and RNA-seq of pupil wings, as well as HCR RNA fish of these pupil wings to really get at whether optics is in fact in charge here of producing this AFP. And also, Sean just got back from a trip out west, collected over 80 butterflies, hybrids of lorquini and Weidemeyeri, and so we're going to be able to, in future studies, look at the look at this AFP extent as a quantitative trait instead of just a binary trait, and start to disentangle the genes that are involved also in regulating the size of this AFP. Lots of folks to thank in the Mullen Lab as well as collaborators, and I want to, before taking any questions, just say that we're also looking for a postdoc to join this project. We're going to have lots of new and exciting data, so if anyone out there is looking for an exciting postdoc at Boston University, email me or Sean, and I'd be happy to take any questions. Yeah.
Great job. If, I don't know a lot about this system, so it's going to be a dumb question, but if there is two species, one of them isn't mimicking really well, and they can hybridize with the other, do we know what kind of selection pressures might be kind of keeping the hybrid from taking over the other non-mimic original phenotype?
Yeah, so why don't the hybrids become dominant in the system?
Since they like can mimic a little bit, are they just not in the environment where that mimicking is useful?
Yeah, that's really interesting. I would imagine that changes based on how common the model is as well in the system. So I don't have a very clear answer for you, but I will say that it does really matter the size of that AFP for kind of bird predators to be able to see the butterfly and identify it as potentially toxic. And so those kind of any hybrid that's not like a really bright, conspicuous orange patch would probably not have that same kind of benefit. So that could be why those especially lower or smaller AFP hybrids aren't dominating that system. Yeah, cool. I don't think I have any more time, but please find me after, and thank you so much.
All right, well, thanks for sticking around to the last talk before the break. I'm Tim, and I'm actually not going to have any genomic data to show you. I'm going to tell you a story that I think is pretty exciting that's getting started in my lab, so I apologized if you were tricked into staying for this. So I'm at the University of Puerto Rico in Mayagüez on the west side, and what Puerto Rico is experiencing right now I think is pretty amazing. So Puerto Rico is the smallest of the 3 big islands in the Greater Antilles, just about under 9,000 square kilometers, and has about 86 terrestrial reptile species that are native to it. So this is a classic place for studying evolution. We're all very familiar with the Anole story. But that's not exactly what I'm going to talk about. So in addition to the diversity of reptiles that are present in Puerto Rico, there's a lot of diverse habitat. And what Puerto Rico is classically known for are the relatively dry areas in the south and these 3 large mountain chains, the Central Cordillera, as well as the Calle and Luquillo mountain ranges in the east. Lots of classic endemism, high diversity in these mountain ranges, particularly the Puerto Rican coqui frogs, right? But we also have variability in the types of soil that also plays into leading to this diversity, as well as forest cover. So Puerto Rico was completely deforested at one point in the recent past. Basically everything that's there today is secondary forest, besides a few very small stands. And this is going to play into the story that I'm about to tell you. Just keep in mind this diversity of habitat types across the island. We also have very large centers of urbanization that are centered around the coasts, particularly in the northeast, the San Juan metro area, and then kind of south central with Ponce. And then in the northwest, we've got Aguadilla, and if you're traveled to Puerto Rico for vacation, you've probably been to Green Con, which is in the northwest. So, but we have very few protected areas in Puerto Rico that receive total protection from development. So this plays into as well the way that animals get moved around. So I'm going to focus most of this talk talking about snakes. So the native snake species in Puerto Rico are pretty limited. So on the main island, we've only got about 7 species, and they're primarily one of these 3 flavors. So we have several species of blind snakes with kind of a patchy distribution. We have 2 species of racer or colubrid snakes, and then 2 species of the Caribbean boas. So the primary being the endangered Puerto Rican boa, Chilobothris inornatus, and then we also have a single population of Virgin Island boas on the main island of Puerto Rico. But Puerto Rico, throughout its history, particularly since colonization, has also been an island that's been frequently invaded by non-native reptile species. These are usually almost exclusively human-mediated invasions. Some of them can be quite old. In particular, iguana has been in the news a lot recently for spreading all over the Caribbean as an invasive. In the past 40 years in Puerto Rico, their populations have really exploded. But some of the earliest French explorers have recorded notes of seeing green iguanas in Puerto Rico. So it's really unclear if there's also sort of natural invasions that have occurred over time, and the populations never really got established because there was another resident already, the rock iguana. So the Puerto Rican rock iguana is now restricted only to Mona Island, far in the west in the Mona Strait. But we have an invasive population of Cuban rock iguanas in Puerto Rico due to an accidental release from a zoo on the island of Magallas in the 60s. So now, interestingly, not going to talk too much about this, green iguanas are also invading Magallas Island, where they're interacting a lot more with Cyclura, with the Cuban rock iguanas. And so there's some interesting things to look at there. For about the past 15 years, there's been one established population of Xenocrophis from Southeast Asia, probably, again, a human-mediated introduction because of trade. We have several species of invasive geckos. A couple are completely widespread across the entire island, and then we've got large tokay geckos, gecko, in just a couple of locations. And then since the 50s, we've also had the global invader, the red-eared slider. But what's actually going on now is we have an invasion of what I'm going to be calling apex predators, because when we think of the native snakes that are found in Puerto Rico, Chilobothris inornatus, the Puerto Rican boa, is literally like the apex predator on the island. So the only things that eat very large boas on the islands, in Puerto Rico anyway, would be domesticated animals, birds, and then on other Caribbean islands that have crocodilians, they would be a big predator of large snakes. Puerto Rico, it's pretty much restricted to the domestic animals and some birds. But since the 1990s, boa constrictors from, well, I'll talk about where from in a minute, were introduced in the west side of the island in Magallas due to another release, accidental release from zoos. And so they've since been spreading. They've been established since the mid-90s, but in the past 10 years, they've been very rapidly spreading, interestingly enough, along with green iguana populations. About 10 years ago, 10 to 12 years ago, the first reticulated pythons were, from Southeast Asia, were spotted in the wild in Puerto Rico. These were originally assumed to just be accidental, illegal pet releases. I should note that Puerto Rico has very strict laws on keeping reptiles as pets, and so the ball python in the bottom right corner is actually the only snake of these that is legal to have as a pet in Puerto Rico. So boas, retics, if they're being found and they are escaped pets, they're illegal escaped pets. And so moving on, breaking each one of these snakes down, so boa constrictors, the red dot indicates the assumed or known points of origin. Black dots indicate established populations. And so in 2014, Graham Reynolds and my collaborator Alberto Puente and others did some haplotype analysis and genotyping and determined that there was a single haplotype at that time on the island, and it was most similar to boa constrictor from South America, Brazil specifically. And so typically in the wild, in their native ranges, boa constrictor, their diet primarily consists of small mammals, some birds, and lizards. In Puerto Rico, they're eating many things, shifting their diets a little bit. And I just want to point out that one of the diet items that has been found in invasive boa constrictor populations by my collaborator Alberto is the endangered Puerto Rican parrot. So they're also eating endangered species. As far as the data on the exponential increase, so in 2019, from a single DRNA facility where these boas are often turned in, or the Puerto Rican DRNA gets called, they go out and they pick up a snake, they bring it back to this holding facility. 2019 and leading up to 2019, there was typically less than 100 per year. So far in 2024, there's been more than 400 from that single facility. Again, they're impacting endangered species. I threw this picture in. I'm not sure if Warren is in the crowd, but interestingly enough, there's also some like weird genetic morphs that come out of the wild population that the pet trade people are really interested in too. So like these striped individuals. So, but what's really interesting with the boas is if you notice this spread pattern, they're sort of following the coasts, following the typical like forested areas that you would expect. But there's one population in the very southwest tip of Puerto Rico that in the Cabo Rojo National Wildlife Refuge, or Las Salinas, that they're in a totally salt marsh environment. This is totally different than any native habitat for any native boa constrictor, or even boa constrictor imperator in Central America. And so some of the individuals that we're getting from this particular location are phenotypically a little bit more similar to boa constrictor imperator. I just have to shout out to one of Alberto's former graduate students, Fabian Feliciano. He collected a lot of the boas, particularly from this area. So this has consisted of daytime and nighttime surveys 5 days a week ever since last December. Capture rates have not gone down, and they're already have already removed, and they're removing every single animal in this case. Over 300 animals have been removed, in 8 months from a single 5-kilometer transect. And so what we see in these animals, that the body size is actually shifting. So this is an adult female from the Las Salinas area down on the left. This is a picture of a typical adult female boa constrictor that was captured from a forested area on the top right. That's a 6-foot table. I mean, I know it's skewed a little bit for the distortion, but it was an over 8-foot snake, which is much more typical for boa constrictor. And so again, over 300 individuals from this one particular 5-kilometer site, and we're observing shifts in the type of habitat they use, shifts in body size, drastic shifts in diet. They're almost exclusively only eating lizards there. And then there's some preliminary data about that they're shifting like their activity time. And one thing about the habitat shifts is, interestingly, those much smaller boas in this population, during the dry seasons, they're always in the trees, which is a little opposite what I would respect. And during the wet season, they're always in the ground where it's like flooded in very saline water. So physiologically, what's going on, I'm particularly interested in. So I'm going to speed this up a little bit. We also have 2 invasive populations of reticulated pythons that have been spreading. So the red dot is the original source population. Some of those individuals were captured, moved to a holding facility more in the center of the island. Subsequently, some of those escaped and established a second population. So reticulated pythons are the longest snakes in the world. Some have been measured at close to 30 feet. So far in Puerto Rico, our largest one is 4.9 meters. Should go back. So this is my colleague Alberto and a couple of his students doing a dissection on one of the big ones. And so some interesting life history stuff. It doesn't seem to be tied to a clear reproductive season. In Puerto Rico, we're finding juveniles and eggs like kind of throughout the year. Even very morphologically distinct, phenotypically distinct things like albinos. Just like with the boa constrictors, these morphs that are, usually would be selected out by predators, is what the assumption is, they're persisting because there's really nothing that's attacking these. And so I just want to also give a shout out to this gentleman, Wilson. He's running this project to help us get a lot of fine scale collections of these reticulated pythons. So this is just a plot of all the snakes he's collected just in the past 6 months. This one was collected yesterday after it ate someone's cat. So that's a cat in the belly that he's holding up. Moving on to the ball pythons, there's one established population in the west, again in the city of Mayaguez. We believe it's established due to the fact that we're finding juveniles, we're finding females, with eggs, about to deposit eggs. But again, all of these individual snakes came out of the wild. Now, certainly some of these probably are recently escaped pets, but there's so many in this particular area that we're pretty confident that they're also reproducing. So all of these morphs aren't being selected out. So some of the risks that these snakes present and some of the interesting things. So this is my grad student, Jacob Adam. He's been working with the Puerto Rican boa and the fungal microbiome. These are some of the caves that he's been focusing his work on, where the snakes use the caves. We haven't found any invasive boas in caves yet, but they're all in the same forests. And we are finding ophidiomyces in some of the caves, so that's the causal agent for snake fungal disease. This is another student of mine, Veronica Batto. She's been looking at the invasive boa populations and screening for reptoranovirus, which causes inclusion bodies and inclusion body disease, 100% lethal disease of snakes. There's just a slide with some inclusion bodies in the blood. So some of the ideas that we want to do some population genomics, really look at adaptation, especially in these boa populations that are invading these novel habitat types. I do a lot of microbiome stuff. I'm really interested in metagenomic interactions. Again, community-wide effects. There are also invasive caimans. So far, only the reticulated pythons occupy an area where there's also caimans, so it's kind of interesting. There may be some predation going on, particularly there. And then this is just like some of our sampling so far. So 21 different types of tissues and microbial samples from a lot of snakes. Need to do more sampling on the ball pythons. But yeah, just want to acknowledge NSF and thank everyone. I ran out of time for questions and I thought I would have plenty, but catch me later.